Whenever parasites infect humans, humans always have its own way of preventing the progression of infection. Thus, in each infection of parasites, we have a host immune response against these parasites. And in this topic, we'll be discussing on how our body would try to get rid of these parasites in hopes of preventing the further progression or further multiplication of these parasites. The following are the required readings for this topic. It's from Chapter 1 from Belisario, Chapter 1 from Clinical Parasitology, and Chapter 1 from Essentials of Parasitology by Sastry. We start with a trigger case. We have a 21-year-old sexually active college student who visited the student's clinic due to a 2-day onset of vaginal pruritus associated with discharge of foul-smelling, greenish frothy vaginal discharge. Now, this case is a very classic presentation of Trichomonas vaginalis infection or Trichomonas vaginitis, as evidenced by the presence of greenish, frothy vaginal discharge that is very typical of Trichomonas vaginitis. And the most common clinical manifestation of Trichomonas vaginalis is vaginal pruritus or vaginal itchiness. Pruritus means itchiness. Now, for this case, what are the initial protective factors against the parasite causing this patient's condition? Now, to answer this, we have to discuss how our immune system works and how is it uh, organized. The immune system is divided into innate immune system and the acquired immune system. The first line of defense would always come from the innate immune system, and the innate immune system is divided into the physical barriers, which basically makes up the frontliner of our immune response. And it would comprise our skin, our mucous membranes, saliva, the flushing action of urine and tears, and stomach acid. And all of these physical barriers aims to stop infection before it enters the body. Our innate immune system also include blood-borne products such as the phagocytes and complement cascades. Now, phagocytes are part of the formed elements of blood. Remember, our blood is comprised of two components. We have the plasma and the formed elements. In the formed elements, we have the erythrocytes and the leukocytes. And then leukocytes, we have the phagocytes, the neutrophils. We have the monocytes in the blood, it's called monocytes. In the tissues, it's called macrophages. Remember that macrophages originates from monocytes. Again, monocytes, these are blood-borne phagocytes, while macrophages, they are already deposited in the tissues. Now, aside from the macrophages and monocytes, we have the basophils, eosinophils, and natural killer cells. All of these cells aim to cause death of dangerous microorganisms, while the complement cascade, uh, it also results into the direct killing of microorganisms or can also activate or can cause chemoattraction of the phagocytes. Now, our acquired immune system is divided into two as well. Okay, we have the B cell immunity, also known as the humoral immunity. This is the antibody mediated immunity. The humoral immunity is also known as the antibody mediated immunity, and the T cell immunity, which is also known as the cell mediated immunity. Now, both of these both branches of the acquired immunity are interdependent with each other. The B cell immunity is dependent on the T cell immunity because activation of your humoral immunity requires you to have activation as well of your cell mediated immunity. Okay? Now, in terms of parasitic infections, we have two major factors in the immune response. We have the host factors and parasitic factors. Host factors would include immune status, age, underlying disease, or several comorbidities the nutritional status of the patient, and the genetic constitution and various defense mechanism of the host. In terms of parasitic factors, very important to note is the size and the tumor burden, the route of entry, the frequency of infection, parasitic load, and various immune evasions of mechanisms of parasites. Now, as what I've previously mentioned, it is an interplay between host and parasitic factors in terms of how you become prone to become infected with a parasite. Now, it doesn't necessarily mean that if you are infected with if you ingest one ova, that means that you'll be definitely infected with a parasite. Now, it still depends on multiple factors. It still depends on how many of that ova have you been consuming or have you been ingesting, or in terms of your host factor, how well your immune response would try to attack the further progression or the further development of these parasites. So it's actually an interplay. It's not a hard and fast rule that if you ingest one ova of the parasite or one infective form of the parasite, it doesn't always mean that you become infected. So we have several outcomes of parasitic infection. The first is parasite fails to become established in the host because it can be due to a multiple it can be due to multiple factors. It can be due to the low tumor burden or the low parasite burden or the low parasite load. It can be also to the inactivation of the parasite by our innate immune system, or it can be inactivation of the parasite by our acquired or adaptive immune system. Second, parasite become established and the host eliminates the infection. Now, the parasite is successful in establishing or in fulfilling its life cycle within the host, but the host is also successful in eliminating that parasite. The third outcome is that the parasite becomes established and the host begins to overcome the infection but is not totally successful. The fourth outcome is the parasite becomes established in the host in trying to eliminate the organism but becomes damaged itself. So in this type of outcome, the host aims to kill the parasite. But during the process of killing the parasite, the intense immune and inflammatory response against the parasite also causes damage to the normal or the neighboring normal tissues where that parasite becomes deposited, right? And lastly, parasite becomes established and kills the host. This is the most deadly outcome of infection. Now, the innate immunity is the resistance which an individual possesses by birth. It can be due to a genetic and constitutional makeup. Now, there are several factors that could influence the innate immunity. These are the age of the host, the sex, nutritional status, and the genetic constitution of individuals. A very good example of this one is that in people with hemoglobin S or those who have abnormal hemoglobin, now remember hemoglobin consists of two alpha and two beta subunits. But in sickle cell disease, you have an abnormal production of your beta subunit of hemoglobin. So if you have an abnormal production of beta subunit, and the reason for that is because of a point mutation in the beta globin gene, it can result to what we call a formation of hemoglobin S. No, the normal hemoglobin should be hemoglobin A. Now for hemoglobin S, this can predispose a person to develop sickle cell disease. Okay, and also those individuals with fetal hemoglobin and thalassemia hemoglobin, they are actually resistant to falciparum malaria. So those individuals who have sickle cell disease, they are inherently resistant to falciparum malaria or the severe type of malaria. We have several types of malaria, and Plasmodium falciparum is the most severe type. Is the parasite that causes the most severe type of malaria. Now, sickle cell disease is somewhat the this resistance of individuals with sickle cell disease is somewhat an evolutionary advantage to that of falciparum malaria. Now, the in Africa, where there's the highest incidence of malaria, it is also where you can find a lot of individuals with sickle cell disease. So therefore, for the longest time in their history, they have been exposed to malarial infections. It became an evolutionary advantage for those individuals with sickle cell disease in an area wherein you have a lot of malaria. So it is quite a protective factor against them. Now, for those individuals who have Duffy blood group negative red blood cells, they are also resistant to Vivax malaria. Okay, so this is what I've been telling you. So this is an evidence of what I've been telling you. So we have resistance to Plasmodium falciparum in sickle cell trait. Erythrocyte is driven by oxygen-dependent growth inhibition. Now, those individuals who have sickle cell hemoglobin or hemoglobin S, they have this mechanism in which they would resist the infection to Plasmodium falciparum, which is the causative agent of the severe type of malaria. Now, in this research, they have pointed out that polymerization of sickle hemoglobin is responsible for growth arrest of the intraerythrocytic Plasmodium falciparum parasite. And the polymerization-induced growth inhibition of these parasites following cytoadherence is the critical driver of the reduced parasite densities observed in malaria infections of individuals with AS. AS stands for sickle cell hemoglobin. And this protective effect of sickle cell hemoglobin derives largely from effective sequestration of infected red blood cells into the hypoxic microcirculation. Now, what does this research tell us? Now, this research tells us that uh, remember that Plasmodium falciparum, they infect red blood cells. And what type of protein, what unique protein is usually found within the red blood cell? It's the hemoglobin, right? And the hemoglobin, it sorts of uh, function as a food to the Plasmodium falciparum. Now, if you have an abnormal hemoglobin, it causes polymerization, and this polymerization will inhibit or will make the Plasmodium falciparum accessible to consuming that hemoglobin. Therefore, it will cause retardation of the growth of parasites. Therefore, you have this somewhat an innate resistance to Plasmodium falciparum if you have hemoglobin S or sickle cell hemoglobin. Now, what are the several components of innate immunity? As what I've previously mentioned, we have the anatomic barriers such as the skin and mucosa. They form as physical barriers. And skin is an important barrier for the parasite that enters the cutaneous routes, such as the schistosomes, hookworms, and strongyloides. So these three parasites, they infect humans through skin penetration. So these three parasites are important for skin penetration. Now, physiologic barriers include pH, uh, pH, and very soluble molecules such as lysozymes, interferons, and complement proteins. Now, gastric acidity in our stomach acts as a physiologic barrier against Giardia lamblia and Racunculus medinensis. Now, in cases wherein you have low gastric, wherein you have um, low levels of gastric acidity, therefore your stomach is more alkaline, you are more prone to multitude of infections, can be bacterial and parasitic infections. Now, if you have an alkaline stomach environment, it easily allows passage of our cysts such as the Giardia lamblia cyst, and easy passage of Giardia lamblia would eventually develop into the encystation of the parasite that will eventually form the infection. That will definitely result into the infection. Now, phagocytosis is also a component of innate immunity. Now, phagocytes like macrophages and microphages, such as neutrophils, basophils, and eosinophils, acts as the first line defense against the parasite. Now, among all of these phagocytes, it is the eosinophils that is the most important against parasitic infection, and especially for helminthic infection. Now, if we have elevated levels of eosinophils in the blood, also known as eosinophilia, it points out or it gives you a clue that the patient may be having a parasitic infection. But that is not true all the time. That wherein you have an elevated eosinophils in the blood, it doesn't mean that you have parasitic infection. It can tell you, it suggests that this patient may have a parasitic infection, but that doesn't necessarily mean that it is always parasitic infection. Now we have other several cases wherein you have elevated eosinophils, but the patient does not have a parasitic infection. So example of that is the acute interstitial nephritis. Now, acute interstitial nephritis is a type of acute kidney injury wherein it's characterized to have increased levels of eosinophils in the blood as well as in the urine. So again, what I am emphasizing is that in parasitic infection, usually or typically, you have elevated levels of eosinophils in the blood. But that doesn't necessarily mean that if you have high levels of eosinophils, it always points out to a parasitic infection. It can also tell you other types of etiology such as acute interstitial nephritis, which is a type of acute kidney injury. Now, for complements, they play an important role for killing extracellular parasites by forming a membrane attack complexes that would eventually lead to the formation of holes in the parasite membrane. Now, natural killer cells, they are another important mediator of innate immunity, and they play a central role in killing few of the helminthic parasites. Now, how does our innate immunity initiates further immune response? Now, we have this type of receptor. This is a very important receptor, the toll-like receptors. They are type 1 integral membrane receptors. And what do? Since they are receptors, they receive something, right? Receptors, they are membrane-bound proteins that receives signals from the outside. They receive something from the outside telling you that, hey, we may have an infection and we need to initiate our immune response. So that is the role of the toll-like receptor. Just like uh, in super highways or if you have been to major highways in Metro Manila, they have toll. So it's like you are entering to a road and it will signal our cell that our body that we may have uh, we may have an a progressing infection. So what do they do? Toll-like receptors, they recognize specific molecules called pathogen-associated molecular patterns or PAMPs that are non-native to the body. So where do these PAMPs come from? They basically come from the microorganism. For example, from a parasite or a bacteria, it's like a certain portion of their part of their bacteria or the parasite or even the metabolic products of that parasite. So these are the pathogen-associated molecular patterns. Now, toll-like receptors recognize these pumps. Okay, they are basically the signals. They are the signals that tells our body that hey, we have an infection. So it is the role of the toll-like receptor to recognize these pumps so that it can initiate our immune response. Okay? Toll-like receptors are expressed on various types of cells, and we have 10 functional human and 12 mouse toll-like receptors that have been identified and is currently um, is an active area of immunologic research. Okay, so toll-like receptors, they are initiators of immunity, and we have several types. Now, toll-like receptors types 1, 2, 4, 5, 6, and 10 are expressed on the surface of the cell to recognize extracellular pumps, while toll-like receptors 3, 7, 8, 9 are expressed on in compartments such as the endoplasmic reticulum, endosomes, and lysosome. Basically, they try to detect intracellular pumps. Again, where do these pumps come from? They come from the microorganism. They would try to detect a sample of that microorganism so that our toll-like receptors will initiate a cascade of information telling our immune system that hey, that we have an infection. Now, if a pump or if a pump attaches to a toll-like receptors, it will now initiate several signal transduction cascade. Now, in TLR signaling pathway, we have two major pathways. We have the TRIF pathway and the MYD88 pathway. So I'm not expecting you to remember all of these things, but the main point of this is that if a toll-like receptors recognize a pump, it activates this signaling pathway, and eventually the end product of the activation of this pathway is the upregulation or the synthesis of cytokines such as interferons and some transcription factors such as NF-kappa B and the AP1. So these interferons, the one that you see here, this one, the interferons, they are cytokines. They would promote further activation of our innate and as well as the adaptive immunity. So they will send a signal now to our T cells that hey, we have an underlying infection. You have to become activated. Now, our NF-kappa B and AP1, they also promote further upregulation of pro-inflammatory cytokines or those cytokines that will promote inflammation and activation of the immune system. Now, to further dissect through the mechanisms of the toll-like receptor signaling pathway, the parasite ligands activate toll-like receptors and mediate signals through either MYD88 or myeloid differentiation factor 88 or toll interleukin-1 receptor domain containing adaptor-inducing interferon B pathways or TIR pathways. Now, toll-like receptors initiate inflammation through the activation of NF-kappa B and other transcription factor. So basically, it is a pro-inflammatory signaling pathway. It promotes inflammation. And why do we want inflammation? We want inflammation to kill the parasite. That is the end goal. And this activation of the activation of transcription factors, it would result into the subsequent production of pro-inflammatory cytokines and production of reactive oxygen and nitrogen intermediates. Now, based on your understanding, the previous understanding about reactive and nitrogen intermediates, these substances or these species, reactive oxygen and nitrogen species, they are highly active. They can easily kill microorganisms, even our own cells they can kill. Okay, so for reactive oxygen species, it is also a mechanism to kill microorganisms such as parasites. TLRs activate antigen-presenting cells and bridge innate and adaptive immunity by coordinating response of the T cells and B cells. So this is what I've been mentioning. The toll-like receptors will now activate the antigen-presenting cells. And these antigen-presenting cells will get a sample of the parasite. Since from the name itself, it gets a sample of the parasite in the form of an antigen, and they will now present it to the acquired immune system. And if they now present it to the acquired immune system, it now forms your adaptive immunity against that microorganism. So that is how our TLRs are very important in bridging innate and adaptive immunity. Okay, so some parasites, for example, Leishmania, Entamoeba, and Trypanosoma can downregulate TLR expression and it is an important part of an immune evasion strategies. Now, the previous slides I've presented that in parasite factors, immune evasion mechanism can also play a role in the infectivity of a parasite. Now, these parasites that are flash leishmania, entamoeba, and trypanosoma, they can downregulate TLRs. So, what do you think is the implication if you have downregulation of TLRs? So, if you have downregulation of TLRs, meaning you have lesser capacity of recognizing pumps. If you have lesser capacity of recognizing pumps, then you have lesser capacity of activating your immune system and lesser activation of the inflammatory cascade. So, no one now will protect your body from this infection. So, that is why these parasites are also important because they can evade our immune response through downregulation of the toll-like receptors. Now, I want you to be familiar with the several TLR agonists expressed by different protozoans and some helminths. Just familiarize yourself. I do not expect you to memorize all of these, but please be familiar with the parasite molecule and what type of toll-like receptor that is being activated. So, for example, if a patient is infected with the malarial parasite Plasmodium falciparum, so what are the pumps that is detected by our TLR? For example, if it's TLR9, it's from the Plasmodium hemazoin. Now, the Plasmodium hemazoin, this is a byproduct of the metabolism or the digestion of our Plasmodium of falciparum. This is a byproduct of their hemoglobin digestion because, as what I previously said, Plasmodium likes to eat hemoglobin, and their digestive byproduct or end product can be a hemozoin. So that hemozoin will now form as a pump, okay, or pathogen-associated molecular pattern that is being detected by our toll-like receptors. Now, some genetic material of our Plasmodium DNA is also capable of being detected by our toll-like receptors. Okay, so what is the therapeutic role of toll-like receptors in parasitic infections? So TLR agonist or antagonist of their negative regulators play a novel therapeutic role for parasitic infections or as adjuvant for vaccine as in malaria and leishmaniasis. Interference with TLR function is likely to prevent their aberrant activation and excessive release of inflammatory mediators. Now, remember, in inflammation, we do not want excessive inflammation. We do not also want uh, a substandard or a sub-level of inflammation. We just want a balance between high and low inflammatory response. We do not also want excessive inflammation. And why do we, and what is the implication if we have excessive inflammation? If we have excessive inflammation, as what I've previously said, it can promote further tissue damage of the normal cells surrounding the parasites or even our entire body system. If we have too much of inflammatory response, if our inflammatory response is exaggerated, then that also poses a critical damage to our normal tissues. Now, in some parasitic infection in which you have excessive inflammatory response, we can also play through the toll-like receptors. We can downregulate the toll-like receptors, an example of which is the E6446, which is a small molecule that acts as an antagonist for nucleic acid sensing TLRs, which decreases activation of TLR9 and reduce the levels of pro-inflammatory cytokines. Okay, so for this one, it can be a potential drug for cases of malaria. Because in malaria, what is also a problem in malaria is that you have an excessive inflammatory response. Aside from that, of the damage caused by the parasite itself to the host, our excessive inflammation can also produce further damage to our cells. So we want also to balance that excessive inflammation by down regulating. So we can play you can play around our TLRs by creating a drug to downregulate or to inhibit TLR sensing. Okay, so now let's move on to adaptive immunity. Now, adaptive immunity, this is the resistance acquired by an individual during life following exposure to an antigen. It is mediated by antibody produced by the B cells, which is the humoral immune response, or the T cells, or the cell-mediated immune response. Now, it is usually the cell-mediated immunity that is activated first in the adaptive immunity because the cell-mediated immunity will also tell the humoral or the antibody-mediated immunity on what to do next. Now, when a parasite enters the body, the parasitic antigens are processed by antigen-presenting cells, such as macrophages, some of the dendritic cells, and Langerhans cells, which present the antigen peptides to the T helper cell. Now, when the antigen-presenting cells engulf these parasitic antigens, they also secrete interleukin-1 that activates the resting T helper cells. Now, the activated T helper cells differentiates now into Th1 and Th2 cells. Now, for a good visual representation of what I've been discussing, this is the antigen presenting cell in our immune system. Now, antigen presenting cells, they are usually found on the epithelial surfaces, such as the epithelial lining of our skin, in the gastrointestinal tract, as well as in the respiratory tract. Examples of antigen presenting cells are macrophages, Langerhans cells, the dendritic cells, microglial cells. In the liver, it's the Kupffer cells. So these are the antigen presenting cells, and usually they originate from the monocyte. They are monocyte derivatives. Now, what are the functions of the antigen presenting cells? The antigen presenting cells, they somewhat get a sample of the parasite in a form of a parasitic antigen. And the antigen presenting cell, it now presents that antigen to the helper T cells. So they will show or they will present it to the T cells, telling you that, hey, we have here a sample of the intruder. Try to get rid of this one. If you see this type of pattern, you better kill it. So that's the major role of this antigen presentation. Now, aside from presenting the antigen to the T cells, the antigen presenting cell also secrete interleukin-1 that will activate the T cell to form to differentiate into either Th1 or Th2. Now, if the if the T cell differentiates into Th1, okay, it requires these several interleukins to become Th1. It requires interleukin 12. Sometimes it also requires interferon gamma and interferon 2. Oh no, interleukin 2. Okay. Now the interleukins here, they function as signaling molecules to direct the development of the T cells. Now, if it differentiates into Th2, it requires interleukin 4 in order to make a Th2 cell. Now, for Th17, it requires TGF beta and interleukin 6. And for T regulatory cells, it requires TGF beta and FOXP3. Now, we'll discuss individually the Th1 and Th2 response. Now, what is the implication if we have now the differentiation of your T cell to form a Th1, or what we call the Th1 response? Now, Th1 cells or T helper cell 1 cells, they secrete interleukin 2 and interferon gamma. So, what are the functions of these cytokines, interleukin 2 and interferon gamma? Interleukin 2 activates the cytotoxic T cells and natural killer cells, which are cytotoxic to the parasitic cells. They produce perforin and granzyme. So what does this statement refer to? Who's they? It refers to the cytotoxic T cells and the natural killer cells. Now, if so, in summary, if the antigen presenting cell presents it to a native or a naive T cell, when it differentiates into a Th1, that Th1 cell produces interleukin 2 and interferon gamma. That interleukin 2 will further activate other cells in the immune system, specifically the cytotoxic T cells and the natural killer cells. Remember, the natural killer cell is part of the innate immunity. So there's now the interplay between the adaptive immunity and the innate immunity. And these cells, cytotoxic T cells and NK cells, if they are activated with interleukin 2, they can produce perforin and granzymes that are toxic to our parasites. Now, the interferon gamma, it activates the resting macrophages, which in turn become more phagocytic and release free radicals like reactive oxygen species and nitric oxides that kill the intracellular parasites. Now, what if we have a Th2 response? What if we have a differentiation of the Th2 response? The Th2 cells release interleukins 4, 5, 6, and 10, which are involved in the activation of B cells to produce antibodies, okay? Specifically immunoglobulin E by interleukin 4. Now, remember, antibodies are produced from plasma cells, and plasma cells are derivatives of B cells. Now, before it becomes a plasma cells, it is derived from a B cell. So therefore, when a B cell is activated, it is further differentiated to form plasma cells. And that plasma cell is the one responsible for the production of immunoglobulins. And we have several classes of immunoglobulins. We have immunoglobulins G, A, M, E, and D. The most common of these uh, a class of immunoglobulins is immunoglobulin G and immunoglobulin M. Now, specifically interleukin 4, if the Th2 cells produce interleukin 4 and binds to the B cell, it stimulates the B cell to differentiate it to a plasma cells. Okay, it differentiates the B cells to form plasma cells to form IgE. So therefore, interleukin 4 stimulates B cells to produce IgE secreting plasma cells. And what about interleukin 5? Interleukin 5 acts as an chemoattractant for eosinophil. And eosinophilia is a common finding in viral helmintic infection, as what I've previously mentioned. So that is the reason why we have high levels of eosinophils in parasitic infection, because most of parasites would have a Th2 response. Okay, majority of parasitic infection would tend to be more towards the Th2 response. And in the Th2 response, we have the production of interleukin 5, which acts as a chemoattractant for eosinophils. It attracts the eosinophils towards the site of infection, therefore increasing the levels of eosinophils in the blood in the hopes of getting into the site of infection. So that is the purpose of our interleukin 5 and the Th2 response. Okay, just to give you a brief summary of what I've been mentioning about the Th1 and the Th2 response. When antigen presenting cells gets a sample of the antigen from the parasite, it presents it to the naive T cells. Now, at the same time, antigen presenting cells produce interleukin 1, and that production of interleukin 1 activates the naive T cell to form either Th1 or Th2 response. Now, if it's the Th1 response, we produce interleukin 2 and the interferon gamma. Now, what are the cytokines that induce these subsets? We have interferon gamma and interleukin 12. The immunologic reaction triggered in Th1 is the macrophage stimulation and stimulation of IgG production, and this is usually the default immune mechanism against bacterial infections. Now, in Th1 response, it is mostly host defense mechanisms against intracellular microbes such as bacteria, and also some intracellular parasites like malaria. So for the Th2 response, the major cytokines produced by Th2 is interleukin 4, 5, we have 6, 10, and 13, and the cytokines that induce this subset is interleukin 4. Now, the immunologic reactions triggered by the Th2 response is the stimulation of IgE production and activation of mast cells and eosinophils. Again, the chemoattractant for eosinophil is interleukin 5. And it is this Th2 response is a major host defense mechanism against helmintic parasites. That's why you have eosinophilia if you have a helmintic parasitic infection, because of the induction of or the production of interleukin 5 by the Th2 cells. Now, the Th2 response activates B cells to produce antibodies, which in turn have various roles against a parasitic infection. They are first the neutralization of parasitic toxins, which is mediated by immunoglobulin A and immunoglobulin G. Now, immunoglobulin A is the secretory type of immunoglobulins that is usually found in secretions such as the epithelial lining, mucus, sweat, tears, and saliva. It also prevents attachment of the gastrointestinal tract, which is mediated by the secretory immunoglobulin A. And also, these antibodies would agglutinate the parasitic antigens, thus preventing invasion, which is usually mediated by immunoglobulin M. And immunoglobulin M is the largest among all the antibody classes because it is a pentameric structure antibody. Okay. So apart from that, the antibodies also induce complement activation. For this one, the complement binds to the Fc portion of the antibody coated to the parasitic cells, and the activation of the complement leads to membrane damage and lysis, thus killing the parasite. Now, the antibodies can also induce this type of mechanism. Very unique is the antibody-dependent cell-mediated cytotoxicity, ADCC, which is important for killing helminths. Now, for this one, the activation of natural killer cells leads to release of perforin and granzyme that in turn cause membrane damage and lysis, as what I've previously mentioned. Okay. Also, antibodies, specifically immunoglobulin E, antibodies coated on mast cells, when they get bound to parasitic antigen, the mast cells become activated and release a number of mediators like serotonin and histamine. And please do take note that these substances, especially histamine, that is responsible for the production of the sensation of itch or itchiness and swelling, because histamine causes vasodilation, and it causes the irritation of the free nerve endings that you will perceive as an itch. So that's why we usually treat itchiness with antihistamines such as cetirizine, loratadine, desloratadine, and other types of antihistamines. Now, immune response may be exaggerated or inappropriate in the sensitized individuals on the re-exposure to the same antigen. Now, if you have been exposed previously with a parasitic infection, the second time around that you become infected, you may have an exaggerated or too much of immune response, and that is also detrimental. And that type of reaction wherein you have an exaggerated immune response upon re-exposure, or if you are exposed again to the same parasite or the same microorganism, that is what we call hypersensitivity reaction. And this is important because it can be harmful to the host, causing tissue damage, especially to the normal tissues surrounding the parasite or the infectious agent. Okay, now we have four different types of hypersensitivity reaction. The first hypersensitivity reaction is the type 1 hypersensitivity reaction, and these are the allergic or anaphylactic reaction. And this occurs within minutes of exposure. This is quite quick. If you are re-exposed again to that antigen, immediately a reaction can be produced. And this is mediated by immunoglobulin E Okay, the antibody is mediated by immunoglobulin E. It causes degranulation of mast cells. Now, immunoglobulin E, these are usually found on the surface of the mast cells. Now, if these immunoglobulins that are already bound to the mast cells at, binds to an antigen, or an antigen binds to it, it causes degranulation of mast cells. When you say degranulation, it releases whatever intracellular contents of the mast cells, including serotonin, histamine, and elastase and tryptase, which are damaging to the surrounding area. Okay, so it will result now into this anaphylactic or allergic reactions. So examples of this one is the cercarial dermatitis in schistosomiasis, for Loeffler syndrome in ascariasis. Now, Loeffler syndrome, this is a pulmonary this is a pulmonary eosinophilic pneumonitis. It is an allergic reaction in the lungs wherein you have a lot of eosinophils in the lungs because of the migration of the larva of Ascaris, hookworm, or Ascaris hookworm or strongyloides. Now, ground itch caused by hookworm infection, anaphylaxis due to the leakage of hydatid fluid, and Echinococcus granulosis or the dog tapeworm, Cassoni test, which is done to the diagnosis of the hydatid disease, and tropical pulmonary eosinophilia, which is caused by occult filariasis. So again, type 1 hypersensitivity reaction. The reaction is very quick, within minutes or even within seconds of exposure. And this is what we call the allergic or anaphylactic reaction. It is mediated by IgE, causing degranulation of mast cells. Next, we have the type 2 hypersensitivity reactions. These are mediated by immunoglobulin G, or rarely by immunoglobulin M antibodies, produced against the antigen on the surface of the parasitic cells, causing antibody-mediated destruction of the cells by either complement activation or by the NK cell activation through the ADCC. So, as what I've previously mentioned. Examples of type 2 hypersensitivity reaction is anemia in malaria, the black water fever in malaria following quinine therapy, myocarditis in Chagas disease, and the killing of helminths by NK cells. So what are the mechanisms in these examples? Basically, we have antibodies that are produced by our immune systems. Now, it's supposed to kill the microorganism, the parasite, but it can also damage the surrounding cells. The binding of that antibody can also cause damage to the surrounding cells, normal cells of the host, thus inflicting damage. So that is type 2 hypersensitivity reaction. Now, for type 3 hypersensitivity reaction, these are immune complexes that are formed by the combination of parasitic antigens with a circulating body, which then get deposited in various tissue. So how is it different from type 2? In type 2, you have antibodies that binds to tissues or to the parasite at the site of infection. Okay, for type 3 hypersensitivity reaction, we have antigens that are floating around the plasma, and we have also antibodies that floats around that plasma. And in the plasma, in the blood, it is where they form complex. The antibody and antigen form a complex inside the plasma, and when they form a complex, it then gets deposited into various tissues. And when it gets deposited in various tissue, it can cause further damage. So examples of type 3 hypersensitivity reaction is the nephrotic syndrome in Plasmodium malariae. Now, what is nephrotic syndrome? Nephrotic syndrome is a type of kidney injury characterized by the presence of proteins in the urine. So you have now the ability of the ability of our kidneys, the glomerular apparatus of the kidneys are basically damaged. Therefore, it sieves large macromolecules such as protein. Now, normally our kidneys should not filter out proteins, but in nephrotic syndrome, since you have already a damaged basement membrane, you have a damaged glomerular apparatus, Now, large macromolecules in the plasma gets filtered into the urine, and that is why you have nephrotic syndrome. Now, Katayama fever is also a type 3 hypersensitivity reaction, which is the case of in case of schistosomiasis, and also in African trypanosomiasis and onchocerciasis. And the last type of hypersensitivity reaction is the type 4 hypersensitivity reaction. And this is the delayed type of hypersensitivity reaction. Now, this is a T cell mediated. Now, for type 1, it is mediated by IgE. Type 2, it's mediated by IgG or IgM. For type 3, it's also mediated by IgG or IgM, but it forms a complex with the antigen within the plasma and then it gets deposited. But for type 4, you have a T cell mediated. So what happens in here is that when you have a previously sensitized T helper cell, they secrete a variety of cytokines on subsequent exposure to the parasite antigens. Usually, the pathogens is rapidly clear with little tissue damage. However, it may be destructive to the host, resulting in granulomatous reaction. Now, here in type 4, there's no antibodies that are involved. Instead, you have T helper cells that would call on other cytotoxic T cells that will damage the site of infection. So with that, it, it is quite common in type 4 hypersensitivity reaction to form granulomatous reaction. And when we say granulomatous reaction, it is in the site of infection or inflammation, you have a lot of lymphocytes. Why do you have a lot of lymphocytes? Because you have T helper cells. The T helper cells are constantly producing cytokines and interleukins that calls on to other lymphocytes to go into that area of infection or inflammation. Thus, it will elicit a damage to that infected tissue. So that is type 4 hypersensitivity reaction. And examples of these are in elephantiasis, in the cases of filariasis, in granulomatous diseases, in schistosomiasis, and other helminthic infection, and also in cases of leishmaniasis. Now, the parasites can also have some evasion mechanism, as what I have already presented earlier. We have some parasite factors that will try to evade the immune system. And these are the resistance to immune response, immune suppression, antigenic variation, host mimicry, and intracellular sequestration. So read further on the details of these parasite evasion mechanism and how they specifically evade our immune system and what are the examples of parasite that has these mechanisms.